Welcome to this topic on essential and non-essential fatty acids. Fatty acids are energy-rich hydrophobic macronutrients in our diet. They play an important role in metabolism, cell structure, and signaling. In this session, we will present an overview of fatty acids, including their definition, types, classification, and significance of essential fatty acids. I will present the overview of fatty acids. The first question is, what are fatty acids? These are aliphatic carboxylic acid, which are amphipathic molecules with a polar carboxylic group and a non-polar hydrocarbon chain that terminates into methyl group. The most naturally occurring fatty acids have even number of carbon atom, typically ranging from 4 to 24. The names of saturated fatty acids end with anoic, while those of unsaturated fatty acids with enoic. Based on the nature of hydrocarbon chain, fatty acids can be divided into two classes. First class includes saturated fatty acids. They are called saturated because the hydrocarbon chain of these fatty acids is without double bond and is made up of only single covalent bond. Mostly they are solid at room temperature. Palmitic acid is the most abundant saturated fatty acid in nature. Second class includes unsaturated fatty acids. They are called unsaturated because they have one or more than one double bond in their hydrocarbon chain. They are of two types: monounsaturated fatty acid or MUFA, having one double bond in their hydrocarbon chain; polyunsaturated fatty acid or PUFA, having two or more than two double bond in their hydrocarbon chain. Usually they are liquid at room temperature. Oleic acid is the most abundant unsaturated fatty acid in nature. Let us explore the nomenclature of fatty acids. Fatty acids can be named using three different ways or methods. The first one is using delta carbon atom, second is using Greek letters such as alpha, beta and gamma, and the third one is using omega carbon atom. The first two methods are commonly used to name both saturated and unsaturated fatty acids in chemistry and biochemistry, whereas the third method is used to name only unsaturated fatty acids, and this is used more popularly in nutrition. I will explain these methods further using a suitable example of fatty acid. So let us discuss the nomenclature of saturated fatty acids such as palmitic acid. The structure of palmitic acid is shown here. To name any saturated or unsaturated fatty acid, you need three pieces of information. First is number of carbon atom, second number of double bonds, third position of double bonds in the molecule. So let us see number of carbon atom in this molecule, which is obtained using delta carbon nomenclature. Delta carbon starts nomenclature start with the carboxylic end or carboxylic group designated as delta carbon 1. Next will be 2, 3 and so on and so forth. So you can see in this molecule number of carbon atoms are 16. This is a saturated fatty acid, therefore number of double bonds will be 0, and the position of double bond, because there is no double bond here, therefore this point number 3 is not applicable here. This will be applicable only in case of unsaturated fatty acids. So using this information you can write shorthand notation of palmitic acid as 16 carbon colon 0. 0 is indicative of saturation or absence of double bond or saturated fatty acid. The another system of nomenclature which is commonly used in fatty acid metabolism is using Greek letters alpha, beta and gamma. According to this nomenclature, alpha carbon is designated as carbon next to carboxylic group, beta is next to alpha and gamma is next to beta, and so on and so forth. This system I will not elaborate any further in this session. Let us discuss the nomenclature of monounsaturated fatty acids such as oleic acid. The structure of this fatty acid is given here. You need again three pieces of information. First is number of carbon atom, which is obtained using delta carbon system indicating 18 carbon atoms in this molecule. Then second information is number of double bond, which is 1. And third one is the position of double bond with respect to carboxylic group is delta carbon 9. Using this information, you can write down the shorthand form of this unsaturated fatty acids as 18 carbon, one double bond at the position of delta carbon 9. So let us discuss the nomenclature of polyunsaturated fatty acids such as linoleic acid. The structure of linoleic acid is given here. You need three pieces of information to name this compound. First one is number of carbon atom, which is obtained using delta carbon system of nomenclature indicating 18 carbon atoms in this molecule. Second is you need a number of double bond, which is two double bonds, and the position of these double bond in this molecule are at carbon atom delta 9 and delta 12 with respect to carboxylic group. Using this information, you can write down shorthand form of linoleic acid as 18 carbon, 2 double bond at a position of delta 9 and delta 12 carbon atom. Let us discuss nomenclature using omega carbon atom with an example of polyunsaturated fatty acid having 18 carbon atom and 3 double bond in this molecule. This nomenclature is based on the position of the first double bond from the terminal methyl end, which is designated as omega carbon 1, next is omega 2 and 3 and so on and so forth. So as you can see, the first double bond in this molecule from terminal methyl end is at omega 3, therefore it belongs to omega 3 class of fatty acids, and it can be written as 18 carbon 3 double bond omega 3 or alternatively n minus 3. N minus 3 is same as omega 3. It means that the first double bond is present at the third carbon atom from terminal methyl end. This nomenclature is commonly used in nutrition biochemistry. Based on the position of the first double bond from methyl terminal end of fatty acid, these can be divided into three categories. The first one is omega-3 fatty acids that include alpha-linolenic acid, EPA or eicosapentaenoic acid, DHA or docosahexaenoic acid. Second class include omega-6 fatty acids such as linoleic acid, gamma-linolenic acid and arachidonic acid. And the third category includes omega-9 fatty acid including oleic acid. In the next slide, I will present example of each type of fatty acid and their nomenclature. So let us discuss the nomenclature of omega fatty acids using an example of alpha-linolenic acid having 18 carbon atom and 3 double bond. This is kind of polyunsaturated fatty acid. As you can see that the first double bond from terminal methyl end is at omega 3 carbon atom, therefore it belongs to omega 3 fatty acids, and this can be written as 18 carbon 3 double bond omega 3 or n minus 3. The second example is linoleic acid, which also has 18 carbon and double bond, so this is also polyunsaturated fatty acid. As you can see, the first double bond in this molecule from terminal methyl end is at carbon omega carbon 6, therefore it belongs to omega 6 fatty acid category, and this can be written as 18 carbon atoms, 2 double bond omega 6 or n minus 6. The last example is oleic acid, which also has 18 carbon atom and single double bond, so this is monounsaturated fatty acid or MUFA. As you can see, the double bond is at omega 9 from terminal methyl end, therefore it can be written as 18 carbon 1 double bond omega 9 or n minus 9. So these are examples of omega 3, 6 and 9 fatty acids and their nomenclature. Let us discuss the classification of fatty acids based on their nutritional requirements. They are divided into two classes. The first class includes non-essential fatty acids. They are non-essential because body can synthesize them and it is not necessary or essential to include them in the diet. Deficiency of these fatty acids is also unlikely to occur. Second class includes essential fatty acids. They are essential because body cannot synthesize them, therefore they must be included in the diet. The non-inclusion of these fatty acids in diet causes deficiency symptoms. Let us discuss the discovery of essential fatty acids. In 1929, George Bohr and Mildred Bohr showed that rats raised on a fat-free diet developed growth failure, dermatitis, hair loss, and other abnormalities. These symptoms were corrected by adding linoleic acid to the diet, indicating that linoleic acid is an essential dietary nutrient. Later studies identified alpha-linolenic acid as the second essential fatty acid. There are two types of essential fatty acids. The first one is linoleic or omega-6 polyunsaturated fatty acid, and second is alpha-linolenic acid or omega-3 polyunsaturated fatty acids. These are essential as body cannot synthesize them. Therefore, they must be included in the diet. Let us explain why can't we synthesize essential fatty acids such as linoleic acid and alpha-linolenic acid. Normally, unsaturated fatty acids are synthesized by enzyme known as desaturases. These enzymes introduce double bond in the molecule. Desaturases that introduce double bond up to carbon 9 are abundantly expressed in the human body. However, the enzyme that introduce double bond beyond carbon 9 are absent. For example, desaturase 12 that introduces double bond at 12 delta carbon or omega 6 carbon, and 15 desaturase 15 which introduces double bond at 15 delta carbon or omega 3 carbon are missing, and as a result of that human body is unable to synthesize these essential fatty acids, and they must be included therefore in the diet. What is the biochemical indicator or marker of the deficiency of essential fatty acids in humans? In fact, in case of deficiency, oleic acid, which is the most abundant unsaturated fatty acid, is diverted to synthesize meat fatty acid utilizing three enzymes, desaturases 5, 6, and elongase enzymes. As a result of that, meat fatty acid level in blood plasma is increased, which is used as an indicator of the deficiency of essential fatty acids. Meat fatty acid is a 20 carbon polyunsaturated fatty acid known as eicosapentaenoic acid with 3 double bond at carbon 5, 8 and 11. It belongs to omega 9 class of fatty acid. Therefore, by measuring the level of meat fatty acid, one can diagnose or indicate the deficiency of essential fatty acids in humans. Let us discuss the functions of essential fatty acids. Normally, these fatty acids are incorporated into membrane phospholipids, thereby contributing to membrane fluidity, stability, and biological functions. In addition, they are also converted into biologically active long-chain polyunsaturated fatty acids. For example, alpha-linolenic acid is converted into eicosapentaenoic acid and docosahexaenoic acid, which are incorporated into membrane, thereby serving important roles such as cardiac protection, anti-inflammatory, and in the regulation of brain and retina development. Similarly, linoleic acid is converted into arachidonic acid, which is also incorporated into membrane, thereby serving as the precursor of pro-inflammatory mediators, which play important role in tissue protection, initiation, progression, and resolution of inflammation, tissue repair, and healing. However, this conversion of essential fatty acids into these long chain polyunsaturated fatty acid is highly limiting. It is only 1 to 5%. This becomes further limiting in the conditions of the deficiency of essential fatty acids, pregnancy, development, aging, and also certain disease conditions. It become under these conditions important to include these in the diet, and therefore these three EPA, DHA, and arachidonic acid are also known as conditionally essential fatty acids. Let us discuss the sources of essential fatty acids. The first class we discussed is strictly essential fatty acids or essential fatty acids such as alpha-linolenic acid, which is richly present in chia seed, flax seeds, and walnuts, whereas linoleic acid is richly present in vegetable oils, nuts, and seeds. These must be included, therefore, in our diet in right proportion for a good health. Second class, we discussed conditionally essential fatty acids that include three polyunsaturated fatty acids, EPA and DHA. The rich sources of EPA, DHA are fish oil, seafood, as well as breast milk. DHA is richly present in breast milk, which is required for child growth and brain development. Therefore, breastfeeding is promoted or must be promoted. Arachidonic acid, on the other hand, is present richly in eggs, poultry, as well as in red meat and organ meat. Thank you very much for watching this video. Please like it, share it, and subscribe it. Until next time, you take care. Bye bye.